All right. Well, thank you for having me. So it is not often that the head of a small nonprofit gets to speak on the same stage as someone from the World Economic Forum. So I tweeted that out. My company will be very excited. These are the Addgenes that you're looking at. A lot of dedicated scientists and non-scientists, geeks, one and all. We are very proud of what we do. One of the nice things about working in nonprofit is the nonprofit team. So they all get paid, but we're all mission driven, it's quite an inspiring place to work. So I'm going to hopefully bring you some of that inspiration in this post-lunch hour. So if anyone needs to breathe deeply or stand up, please do that. Okay, so Addgene helps scientists share plasmids. We do a lot more now, but that's how we started in 2004. So hopefully everybody in this room understands how publication and material sharing works. You publish a paper, stuff goes in your freezer, no one ever uses it again. And that's a big waste of grad student postdoc time, not to mention taxpayer money, and all the other things that go into reagent sharing. So what Melina Fan, our founder, our scientific founder, she was a grad student at Harvard when she founded Addgene, said, wow, I wrote away for 8 different plasmids. This was before you could make them yourself, of course, before gene synthesis, and nobody got back to her. One person did and sent her the wrong thing. And she's an entrepreneur type. I'm a more run it once it's built type. She's the creative business startup person, and she said, there must be a better way to do this, and founded Addgene with her husband and her brother. And now it's 2016, and I'll tell you about some of the progress we've made since then. So does everybody know what a plasmid is? Okay, plasmid, small circles of DNA, a very good reagent to start with because they're easy to share, easy to store. They're infinitely renewable if you keep them living in bacteria, which is what we do. And they are pretty much the cornerstone of all life sciences research in every organism, in every field, and every experiment you want to do. If you're a policy person, you don't know what a plasmid is, but you know what genome engineering is. Genome engineering starts with plasmids. And so every innovation that we will see in that area has to start somewhere with the plasmid. So it's a small step. There's a lot of other reagents we use, but it's a valuable one early in the process. I think you'll agree. So we think that easier collaboration means better science. So people that request from Addgene get a lot of benefits. They get excellent customer support. So we talked about scaling. So what we've been able to do is have 6 PhD scientists that answer the phone and hundreds of emails every year, just helping scientists know how to clone. Everywhere from undergraduates saying, what's a restriction enzyme, up to postdocs saying, which generation lentiviral packaging vector do I need to use? We have 6 PhDs who are just experts at that. There is no such centralized resource in the world. And if your PI is not a good mentor and can't teach you molecular biology, this helps everybody anywhere get started with molecular biology. You get your materials quickly. I'll talk about how we do that. In the US, it's within days, and we ship to Europe. It's about 4 days it takes to get the materials you requested. Of course, quality control. We sequence everything. We validate what it is. That's unique to science and key for reproducibility initiatives. So I don't have to talk more about that to this crowd. We've shipped to 85 countries. We ship anywhere that UPS allows us to ship. So we shipped to Iran between the war and ISIS incursion, literally the day before the ISIS incursion. We've shipped to a small island off the coast of Spain. Any country where there is academic, nonprofit scientists doing science, we can get plasmids to them. We solve that problem of transport. We do the export and import work. So that's one of the services we provide. And we have a huge amount now of educational resources because we have these smart scientists. We have them share their knowledge with the world openly, and I'll show you a little bit about that. So filling the need supports the mission. We charge for distribution. It's a small fee, on the sly. If you can't afford it, just ask us. We're a nonprofit. We get to do nice stuff like granting plasmids to people who can't afford them in developing countries. We started out really small. We're over 600,000 distributions now, and we're now doing about 100, we'll do 135,000 distributions this year. That's 550 a day to scientists around the world. That's George Church with his granddaughter there. He's a big proponent of Addgene. So having big name people support you in the early years is very helpful. George is a big proponent. So we always say, George, I don't know if you know this, George Church is the reference genome for the world. His sequence, he is the reference genome. So he shares his genome with the world, his genes with his granddaughter, and his plasmids through Addgene. So, and that's really the kind of thing that we're trying to do at Addgene. So we became self-supporting in 2007. We have never had a grant. I have never written a grant. I spent 15 years in pharma, 4 years in for-profit biotech. Now I'm in nonprofit, and even when I was an academic, we had some sugar daddy funding from Hearst. So I have never written a grant in my whole 30-year science career. And here at Addgene, it's no different. We have a beautiful nonprofit model. We charge a little bit for something that we distribute a lot of, and so we are completely self-supporting, not relying on any university or any overriding industrial sponsor. We get to make decisions purely because it helps scientists. We have no investors, we have no stockholders. We completely get to decide because it makes sense for scientists, and that's a hugely empowering thing for someone to be able to do in their job every day. And we really empower the Addgenes to do that. So even when they're on the phone, they can say, "Oh, sorry about that, we'll send it for free." We're like the Zappos or the Amazon of plasmids. So I'll say one more word about scaling, since it's come up a few times and is one of the themes of the meeting. You know, the services we offer and the quality of the service is absolutely directly related to the fact that we got so big. You know, if we were a small repository in one university that had 100 plasmids, nobody would request those. So we have 50,000 plasmids in the database. I don't know if I saw that. Where I'll be talking about depositing in a second. Because we have so much stuff in a central place, we distribute enough stuff to support the resource. And so I'm not going to have time to go into the whole issues with repositories in the world. I hope you know about that controversy in the US and around the world, how they're losing their support. This is a huge problem. We're actually going against exactly what we need to do. Centralized resources that are open, we're killing them. And so Addgene, unfortunately, is above the fray, but we need way more than this to make this work. So why be a nonprofit? I always get asked this. If you're so profitable, why be a nonprofit? So that's the trust. That's the trust part. When we went out and asked scientists to give us their stuff, they don't get any kickbacks. They have a lot of benefits that I'll talk about, but they don't get any money. The university doesn't get any money, but they trust us because we are not a for-profit biotech selling plasmids. Our mission is to accelerate science by helping scientists collaborate. Every time they ask us that, this is what we say. That's how we make decisions. And so that's the trust that allowed the company to get started, and I'll talk about other ways that we dug that trust in to make it be successful. It's also great for employee culture, I have to tell you. I've worked in a very cutthroat, for-profit, crazy biotech, for-profit. Never did I have the kind of energy and excitement in a company as I have at Addgene for people that just want to make a difference. We also pay them pretty well, and they have nice benefits, but that's just an aside. So what we're also trying to do is really, now that we have a little clout in the community, is to make sure that we support open access platforms and educations about scientists. So now we've moved from just the materials in the repository to the knowledge as well, the data about the plasmids. So people say, we don't ship to industry, but actually in industry, all of our sequences are public. Our data are all public. And so you could look at our database and synthesize something in a month, anything that you wanted. So the knowledge keeping is incredibly important. And so we're really proud of the strides we're making there. Take a look at our website. You'll see what the Addgenes do in their spare time, which is write amazing content, best in the world for the website, certainly in the area of genome engineering, stem cells, and some other areas that we work in. So this is some of the educational impact. These are just data from 2015 of our material. These are just the educational resources, not hits on our plasmid pages. And you can see these hot areas, the number of page views, over 1 million in genome engineering and CRISPR-Cas genome engineering. We've had ebooks have now been downloaded thousands of times that have standardized protocols. Again, reproducibility. And so we really are trying to add to this collection all the time. The Science Careers Track is my sideline. It's how we attract people to the website. We give all kinds of advice for scientists, and that's our marketing tool. All the rest is hardcore science content. So we're also a global research. We have a 3-person London office with a Brit, a Spaniard, and a Frenchman from France. So we're a little worried about Brexit, but we're told we could just hang out for a couple years, and they're doing a great job. They're getting around Europe a lot more than we could afford to do it from the US. This is new for us. They've only been there a couple years. We've been solving problems from the US. Now they're helping us solve problems from Europe. And so that's been great. Maybe we'll relocate it if Britain doesn't play nicely with others. But for now, that's working great for us. I can talk about specific countries if you're interested, but we've seen interesting growth. So depositing is free and easy. That's the cool part. What we sell is free because having a good repository is what makes people request, and we make money from requests. So I'm out here getting scientists to deposit. If they deposit cool stuff, we do well, and depositing is free. It's an awesome business model, actually. And so you can see we're up to about 3,000 depositing laboratories. So plasmids have been deposited from all of the 50 NSF-funded research institutions. In fact, we have 700 institutions that deposit. We have relationships with almost every tech transfer office in the world. So also those relationships help us really solve a lot of problems. I would say one other thing about the 50,000 plasmid deposit is it allows us a breadth, again, to serve scientists in all different areas. And they find stuff they didn't know they were looking for, which is another one of the fun things about having a large breadth in one organization. So why deposit? So, you know, first of all, some labs that do a lot of distribution, they have a lot of time. Like there's a tech that's all they do is mail stuff out. It's completely inefficient. We have negotiated excellent rates with UPS. It's much better for us to ship the plasmids to you, aside from the fact that they're the right thing, they're quality controlled. But we also see this intangible set of, and we do quantify it, Cameron, but I think sometimes using a lot of numbers is better than one number. And so they're seeing increased citations because of exposure. People that you didn't know want your stuff, believe it or not. We constantly get emails of scientists saying, wow, I got 10 requests this month. I had no idea anyone was interested in what I did. Incredibly gratifying for the scientists. So, and they have records, by the way, of every one of those. So this is some data that we're generating with our own data now with a collaboration. But for now, these are older data showing that when the red line is when you deposit in a repository, those are your citations. And when you don't deposit, those are your citations. So to me, I don't want to look at the numbers here and Cameron will smack me, but what it says to me is that more people are working together and using stuff. So I'm looking the intangible here, not the number, okay? And that is absolutely what we are seeing at Addgene anecdotally, and we hope to publish a paper this year showing it with the numbers. I'm sure you'll believe me that this is going to be true. So well, how have we solved some problems here? So I love the SynBio community. Everything should be open. Just send it all around. No MTAs. We just want it to be free. That did not work for Addgene in 2004. So our founders took a different approach. Instead, they put a material transfer agreement in place that was absolutely easy to complete in 5 seconds. It's an electronic email. If you're the TTO, the technology transfer officer at your university, you get an email that says, click here, approve this MTA. And so for some organizations, they clear so many MTAs through us that they have to take them out of their statistics because they artificially inflate their effectiveness. There's no redlining. If you want it, you have to click it. I get emails every month from somebody saying, I can't figure out how to download the MTA to make edits. We're like, you want it, you sign it. There is no editing, no lawyer time here. So we actually can't afford to have a lawyer at Addgene now, but he does business development. These MTAs are how they are. They are simple. I'm going to do research with it. If I commercialize, I'm going to come get a license from you first. But other than that, I get to do some science with this. And it happens instantly. We've also even made this faster. It's all electronic, as I mentioned. We have these master agreements that are instantly, the minute the paper is filed, it's done. Nobody has to look at it anymore. How many times have you read the UBMTA? 1 million. You don't need to read it again. Click the button. So a lot of these things are auto-approved if there are no amendments to the MTAs. So we do an MTA on our side when the deposit happens. It's half done. When the requester requests, they complete it. And that's how it happens instantly. And we do, 300 of these a day are completed per day. So then came CRISPR. So here's the case study. So what happened was the early scientists to this field, so Doudna, Charpentier, Church, Zhang, and the other host of scientists working in this area were all committed to open science already. All committed to open science. And they deposited before their papers came out. They said, and their organizations, by the way, supported them and said, yeah, we got a patent fray going on. I have nothing to say about the patent battle. I have nothing to do with it. But we are going to make sure that academic scientists can build on this technology. It's a platform that can do amazing things, right? And so that stuff was there. The day the paper came out, we were like having champagne because literally we're watching the ticker. Feng Zhang's plasmid, purchase. It was that fast. So 3 weeks ago, a Chinese scientist deposited a plasmid on a new technology called Argonaut, for those of you that are aficionados of genome engineering. It is a follow-on to CRISPR, different protein, different system, could be even better. That's the word on the street. So we got a Twitter message that said, oh, try and get that plasmid from the guy in China, ha, like a glove thrown down. And of of course, our scientists are like, challenge accepted. And 3 days later, we got that thing like Shawshank Redemption in the middle of a book, hidden. It's hard. We can get the DNA to them. They have trouble getting the DNA to us. We're working on that. And we put it online. 3 weeks later, it's been distributed 300 times. Okay, so you know that that technology is going to be validated or invalidated very fast. And that's what happens. Would never, ever have happened if it weren't for Addgene. So this is just an example of what's going on in genome engineering. That green is the CRISPR distribution. Those are talons and zinc fingers, some earlier technologies. I thought the scientists might be interested in these data. You can see how much bigger this is than everything else. Genome engineering is cool, CRISPR is miraculous. And we've distributed something like 70,000 CRISPR plasmids since the beginning of 2013. So that's a lot of science. Where are they going? This also reflects our overall distribution. Denmark requests about 0.5% of the plasmids from the repository. You can see where the rest of the things are going. I have to tell you, 0.5%, it's pretty small. So we need to do more marketing here, obviously, in Denmark. But I will tell you why I think the reason is small, and the reason for that is you also do not have very many depositors. So does anybody want to guess, of all the scientists in Denmark that make plasmids in their laboratories, how many of them have deposited to the repository? Not that bad. That's very pessimistic. We have 700 depositors or something like that. I mean, we have 3,000 different depositors from 700 organizations. 17. 20 total deposits. That's pretty bad, actually. Yeah, you should all be going, that's not good. That's not really that good. So there's a lot of science going on here. You know, China is doing better than Denmark. Not that China's not very big, but you have a very collaborative and open, environment, right? You should be depositing better. I have to get up here and say that, otherwise I'm not earning my pay. So please deposit to the repository. So the plasmid collection also, has resulted, as you know, in this technology to a boom in techniques. It's not just genome engineering, it's basic research tools. So Cameron, this is the research, creating the research, right? I'm tagging it, I'm epigenetically studying it, I'm turning it off, I'm turning it on. It's not just the whole ethical issues with human, it's incredibly powerful as a screening tool. So just to give you a few examples, we have over 100 educational resource pages. We're getting a huge number of support. The community writes these for us as well. This one plasmid, which was our most popular plasmid in 2015, so has already resulted in more than 20 published papers. Now, for those of you that know how slow science publication is, you know what what amazing turnaround this is to receive a reagent, then write with it in that short of a time. I actually think this is an underestimate because we can't afford to do the data in the closed access journals because we're a little nonprofit. So we're only looking at open access sources here, or ones that we find coincidentally. Our community often alerts us when they do this. So they're using unsolved questions. We also make available libraries that scientists have deposited. So plasmids are cheap to make, but libraries are expensive to make. We're selling those at cost. It's about $300 for a library to do a screen already made, ready to go in the cells. You can buy it and do your experiment. And so as opposed to designing it, buying it, synthesizing it, whatever you have to do. So we've sold hundreds of these libraries. Papers are already coming out that are very meaningful with results on CRISPR screening. More results. This is another library screen with an Addgene library. Animal work. There's also work done with plasmids requested from Addgene. So these are all examples of the impact, and there are hundreds of them. I'm just giving you a few. This is one of these new technologies, in situ tagging. For the scientists in the room or those that know, this is incredibly, this is like the holy grail of why I don't want to study a gene on a plasmid because it's out of its home in the genome. Now I can study it in the genome in its home place by tagging it easily. Incredibly beautiful experiments are coming out of these technologies. So Jennifer Doudna requested requested plasmids from Feng Zhang and Jonathan Weissman to do work. So she's going to win the Nobel Prize, right, in CRISPR genome engineering, and she's using plasmids from the other people, who, by the way, she's in a legal dispute with. She's requested those plasmids from us and then used them and then published a paper. So that is really, this is one of our favorite anecdotes of like what we can break through by just having people share openly and with safety. So, you know, I think the community realizes the extent to, it's visible in every field, but it's been particularly visible in CRISPR-Cas9, which has been great for us. It's led to a lot of growth and a lot of publicity. So that's fantastic. So we have more resources to do more things. So Addgene is here to help. This is our other tagline. How can we help? And that's what we get to do all the time. I hope I address some of the questions of interest to the organizers of why nonprofit, why scale, why collaborate openly, and why create central resources. And I'm happy to take any questions, and I hope you all deposit if you have plasmids in the freezer. Thanks.
With all this success,
Turn on there.
yeah. With all the success that you have, is anyone ever somewhere in the system tempted to sort of go profit, have an IPO, you know, make some money? Yeah.
Yeah, no. It's so fun. Not at all. One of our founders still works at Addgene. Melina, our scientific founder, is still at Addgene, and there is absolutely no incentive for us to go for profit. Certainly not for me, because I don't have to answer to investors. I don't have to raise money. I don't have to ever raise money. We make money. So it allows me to spend all my time figuring out how to help scientists. And we get a lot of like warm fuzzies from the community. So we get thank you notes, fan mail, you know, go online. We're just sending, if you're on Twitter, you can look at us on Twitter and Facebook. It's a super, super fun place to work. And actually, instead of going for profit, we're taking the mission and we're going to extend it further with the resources we have. So we have a new project going live that will be very exciting in the fall, soft launch in July or August. So that'll be, I think, very impactful. So.
And besides Danish scientists not being good at sharing, are scientists more generally good at this?
So
sharing,
some of them are. So some of them are very good at sharing, and some of them are very wary. And so we have a lot of advocates. So when many scientists become very advocates for Addgene, that's our best. It's better than me getting up here is having other scientists. So often when I go speak, I'll say, are there any depositors in the room? And people will raise their hand and they'll say, it's so great. And I just watch and go, yeah, it's great, you know. So what we really need is the buzz in the community, not us selling it. And that's what's happening is other scientists are saying, how could I not know about this? It's so awesome, you know. And so there are naysayers. I can't express how strongly I feel that openly sharing this type of reagent is a benefit for everyone in every way. It has nothing to do with squelching economics or for-profit development. I worked in pharma happily for 15 years. I believe in drugs. I believe in large companies. We need regulatory. We need big companies to bring therapeutics through regulatory. Academia doesn't do that, Addgene doesn't do that. We need the whole infrastructure. But from the early research, from the basic research perspective, there is no downside to sharing your plasmids, none whatsoever. And so I'm trying to gather we have hundreds of scientists that would say that that's how they feel about it, so.
Questions? Cameron?
So I'm not going to be rude about the numbers because that's a good use of citation as a data source to answer a question which is of relevance to the researchers, and triangulating with usage and citations is a great thing. What I'm really interested in, so you've made your sustainability model is brilliant. So you're taking what is in almost now an informational resource, the sequence of the plasmid, and you're making a sustainability model based on the necessity of physically posting it out.
Right.
Now, how does that play out as synthesis gets? So I'm really interested in this balance between the digital and the physical. Yeah. And
so we did a huge market study on gene synthesis because we wanted to know if we were going to go under. Right? So what if tomorrow you had a machine on your bench that you just put the sequence in and it made the plasmid for you? So we believe we're about 10 years from this technology unless there's a crazy breakthrough. So our follow-on project is to try and protect against that from happening. But actually what's happened with gene synthesis, and I don't have the slides or the data to show you, but we have the data, it's actually spurred Addgene because all the synthesis are making cool tools and plasmids and useful things more than ever before, and someone else wants them. And so yes, if you're here in Denmark, you can probably have that thing synthesized. But if you're in, Nigeria, You need that plasmid. You don't have access to rapid, cheap synthesis. And also for $65 for a sequenced reagent, nobody can beat that yet. We make our own plasmids. It still costs hundreds of dollars and takes a month to get a full plasmid. So when I think it will come, we don't feel like we're at threat right now, and we're shoring up our resources to protect against that because we really believe in the data. The databases are our gem, right? And by the way, all of our data are open. People do research on the data. We don't give it out freely, but we give it out when we have a relationship, because when we give it out, pop-up fake Addgene repositories show up. And we have, I can tell you some funny stories offline about that if you're interested. So, Peter?
Any plans to expand your business here with cell lines, reference compounds, knockout creatures?
Right. So we've done a lot of strategic modeling since I came to the company. It was one of the reasons why I came to Addgene, to help figure out, what was next. So cell lines are tough. They're expensive. What we do is cheap. And so we're preparing to be able to do more types of materials with our infrastructure. We'll be doing some new types of materials in the fall, so you can stay tuned for that. We are very careful and slow about expansion because we do not want to put the very successful core mission at risk. So Addgene is a cautious yet innovative company, I would say. So we do have plans to do more types of materials. One area that we're looking at in particular is standards. Nobody has a better shipment or technology transfer network than we do. So we think we can help a lot in some areas with certain types of materials. And now that people know about us and know our credibility, we're starting to get a lot of offers for that. So we're building out our infrastructure to make that possible. Does that answer your question? I'm not trying to be evasive. Yeah. Cell lines I see is very hard. They're very expensive to keep, and quality control. And we're all about quality, so we get scared by cell lines. Yeah.
Question here?
This is Peter, Novo Nordisk. So I did realize that you didn't want to comment on the CRISPR legal case and the legal battle and patents, but I wondered still if you could reflect a little bit on the the potential liability also from Addgene in shipping CRISPRs to companies, to industry, where commercial licenses may be needed, even though it's intended for research purposes in the first place.
I'm sorry if I wasn't clear about this. The majority of Addgene's library is not available to for-profit requesters. So 98% of our library is only shipped to nonprofit research institutions. A very small portion of the library is available because the depositors are willing to take the responsibility. So Addgene does not take the responsibility. So what happens is, what we do is sort of like scientist A walking down the hall and handing scientist B a plasmid and saying, here, I use this in my lab, you can use it in yours. And the for-profit companies involved in this space understand that that's what we're doing. But if we were to go to the nonprofit understand that, but if we were go to for-profit, there would be that liability and that complication. So if you are at Pfizer and you want one of our plasmids, you have to look at the sequence and make it yourself, or go to the original depositor. And all of our MTAs say that if you want to commercialize, like if you're in academia and you're interested in commercializing, you have to go seek a license. Does that clarify? Yeah. So we stay out of that. Yeah. Yeah? Yes, so we can only safely store materials that are BL2 plus. So anything that goes into the BL4, as you know, we have plenty of lentiviruses, HIV-derived materials, but nothing that needs to be studied past BL2, because our labs are not accredited for those types of materials, and we don't want to be shipping them around. It requires export, import that we can't afford to do. That being said, we have pieces of we have Zika plasmids, we have, all the basic research tools that have been cleared for that safe work. So safety has not been an issue for us. I know in the synthetic biology community, it is a very strong issue, and we align with them in the care that we take. And we've done some work to look at our sequences and make sure. So we have a lot of personal handling. Every deposit is personally handled by a PhD in our company. It's never automated. So we know everything coming in.
More questions? Down there?
My name is Yannick Petersen. I'm from a company called Dare Disrupt. one of the things I've been wondering about, and maybe tying together the morning's presentations, is how do we speed up basic research? How do we disrupt the way we do basic research? And I see what you're doing is actually an accelerator, a tremendous accelerator. So I'm not technical enough to know exactly what you're doing, but I would like to ask you, and maybe the previous speaker, this model of not only like in +1 sharing the results, the outcomes, but now sharing some of the materials you work on, where in other parts of research, and I'm also asking the room actually, where could we copy this model? How could we speed up the research both in Denmark and in Europe? Maybe some people here have good ideas.
Yeah, I would love to hear from the audience. As I mentioned, there are other repositories, but they're often very competitive with one another instead of bringing together. I think antibody reagents is one place that we could be doing better. There's one in Iowa that's actually starting to push that model, but it is centered in a university. It has overhead, it has barriers to growth that we don't have at Addgene. But it is quite an impressive model. So antibodies is one area from the reproducibility perspective, widely used reagents that are poorly characterized and poorly stored, poorly, quality control, and so that's one area. I think there's probably many areas of basic research where you could be doing more collaborative reagent pooling, and hopefully we'll be able to do that as we get bigger. And Cameron, did you want to mention?
Yeah, I think I would just reinforce that. I think it's true right through all of the outputs of research can benefit if they're shared effectively at scale. It's harder for things that are physical. For digital stuff, it's easy. You need digital infrastructures. Sorry, I shouldn't say easy. Relatively cheap. For physical materials, it's more expensive and more difficult. Storage and transport come into play. So I think the spaces where the biggest opportunities lie are where it's very cheap for a researcher to produce or amplify a material. Plasmids are a great case of that, but areas of chemistry and materials, often for a single lab, it's very easy to make a lot of a material. In fact, we often do. And then it sits on a shelf. Centralising that offers some real opportunities. But yeah, as you get to the places where we have the biggest problems with reproducibility, which is where we get the biggest gains, are places like cell lines, organisms, mice, and that gets a lot more complicated. So
one thing I want to say, I should have said, there's a cheap data thing that we could be doing better, and that is protocols. Huge problem. So the paper gets published, the protocol's in there. If there's a mistake, it propagates forever. So a few years ago, a newspaper did a story on my mom on cooking for Rosh Hashanah, the Jewish New Year. And they put 2 tablespoons instead of 2 teaspoons of cloves in the apple cake recipe, okay? It was like, and that is there forever. That's never updated. This is what happens in papers. People make a tiny typo, or the protocol is evolved. So I urge scientists to use Protocols.io and link their protocols back to Protocols.io. Which is, I hopefully will be the up-and-coming way to do this the better way. And I know PLOS is starting to work with Protocols.io and other. So that is one area where we could be doing, because sometimes sharing the materials is not as important as sharing the exactly how to do it. Chemists are pretty good at that, but it's not public. It's not, they have all the steps and the tools, but it's not public. And biologists are notoriously terrible at writing clear protocols. So that's one area that would make a big impact.
Also in journalism.
Yeah.
Anybody else want to chip in on where we could copy-paste this business model to? Or anybody who has a question for Joanne?
Joanne, I just want to, I don't know, now it's working, to challenge you a little bit, because you seem to kind of stay away from the difficult stuff like the cell lines, especially stem cells, knockout transgene animals, etc., etc. I know it's more expensive, I know it's more tedious, but part of the problem we have with reproducibility in this area is because there's not control on what is actually used and given out. So could you comment why you are not entering into this a little more elaborate?
Well, so let me say we're not entering into it first. So Okay, I personally, you know, I think we could do a lot there, and we are building that infrastructure. Again, we are very wary at putting our core mission at risk. So we need to stay under the radar a little bit with things that are contentiously legal. Cell lines become very contentiously legal issues. They're much more valuable reagents. There's more skin in the game. Right? So we're trying to do this in a way with allies that are supporting us, and we're building up those alliances now. So the first thing we do won't be cell lines, but I wouldn't say that in 3 or 4 years we would not be storing some types of cell lines and distributing them through Addgene. So I'm not saying no, I'm just saying not right now. Yeah.
Would that work better in a profit organization, you think?
No, because it would be very expensive. How would the scientists, how would it help them? They wouldn't be able to afford it. Right? So that's one of the problems is financial accessibility as well. So we have to try and keep it cheap, right?
So I wanted to draw, I think this is a really interesting question, and to draw something out that, again, the Commissioner mentioned this in the context of VC funding and scaling across that valley of death. This is a capital problem. So Addgene is undercapitalised to deliver the kinds of things you're asking for. It's not a question of income or revenue, because the revenue model probably plays out, or could be made to play out. It's a question of capital. Our entire innovation and infrastructure system is undercapitalised.
But there was enough capital in the US, wasn't it?
Well, I can't here on mic being recorded tell you how the company was funded for the first 3 years. I will tell you that the founders live with their parents, and so we bootstrapped. So it was really bootstrapped from, and we had good support from Harvard, who gave the founder some space to found the company from one bench and one freezer and one. Fortunately, what Addgene is actually doing is exactly what you say, is building up our capital. So we just built our first BL2 lab 6 months ago. We have, you know, that resource and facility now, so we can actually handle higher cells, not just bacteria, for the first time. And so we are also looking for potential supporters of donations for capital investment. And so we've never taken donations in the whole history of the company, but if we can capitalize enough to be able to have a sustaining model, I think there'd be a lot of bang for the buck.
So maybe you will be. Who knows?
Maybe we will. We don't call it capital at nonprofits. We call it support, donations, stuff like that. We don't.
Sounds so sweet. More questions? Salim, welcome.
Hi there. Given that you've got this amazing view across all of the potential applications, are there 2 areas that you're particularly excited about as to what might result from the work that you're doing and how people leverage it?
2 areas that I'm, wow. You mean of science or of sharing? Like
the application of what you're doing.
The application, yeah. So we do hear about stuff early, and we're careful about confidentiality there. To me, we are not focused on the therapeutic innovations because, which we're very excited about, but we're not focused on that. But we are focused on some of the tools coming out, you know, for research's sake. And so I would say that these the integration tools for the genome that allow you to tag in situ and do in situ tagging, not epigenetic type studies and that kind of thing. I'm very excited personally as a scientist about that. I really think that that's going to change science dramatically on a large scale. And it's going to become the standard. No longer will you be able to throw a plasmid into a cell and expect that to be the last result. It can be a preliminary result, but you're going to have to demonstrate in situ that things are happening. So I think that's a big change for science, and we're moving that direction. And I think also we have a lot of fluorescent tools in the library, and those are becoming more and more sophisticated for diagnostics and you know, basic research. And so those 2 areas, I would say, we're seeing biosensors, a big boom there. Is that enough of an answer? Yeah.
At the back?
I think I might be.
Please wait for a microphone for the recording. And those of you who have questions to ask that
I'll still be here. Yeah.
Cannot be asked when the microphone's on. We'll have a break shortly. You can ask Joanne in the break.
Sure.
Søren Peter Olsen from the Carlsberg Foundation and the Danish National Research Foundation. It's just a comment. Actually, there is another field where similar activities are ongoing, and that's the transgenic animals, the worms, C. elegans. There's a consortium with C. elegans. You can make your transgenic warm, and then it's available for everybody, and it's just a cost price.
So we've been collaborating with all of those fantastic communities, C. elegans, Drosophila, yeast. Those are communities that are very good about sharing amongst themselves already. They don't always have the infrastructure, and when a lab closes up, one of the problems is the archiving of materials that are basically lost to the world, so we're playing a role there. I will tell you that those consortia, both on the material side and on the data side, are losing funding. And in fact, in the US, that's a huge problem. And like even some of the key databases, like the OMIM database, some of you may have used, no funding, losing funding, reduced funding. Because for some reason, NIH and NSF all of a sudden are seeing infrastructure is not as valid as new research. So this is a huge problem. Addgene is not, we are not affected, but all of our community is affected. So it's quite painful to everyone. We try to take whatever we can into the repository to solve that problem. I can tell you one anecdote if you're interested. So I don't know if anybody has ever worked with the Michael Davidson fluorescent plasmid. He's collaborated with hundreds of scientists on his fluorescent reagents. And he called us. When we originally asked him to deposit, he said no, he wouldn't make any money. He wants to sell his own plasmids. We said fine. A few times we were approached by others to get his plasmids, and then he emailed us when I first started at Addgene, and he said, I need to talk to someone about depositing. And I said, okay, and I called him up, and he said, I have cancer. I have 3 months to live, and I have 3,000 useful plasmids in the freezer. And at the time, Addgene had 16,000 plasmids in the collection. So talk about scaling. We actually hired an intern to do the intake on this collection because we knew how valuable it would be to the community. He made it a lot longer than 3 months, but he passed away a few months ago. His whole collection is cataloged. We've distributed thousands of them already. And we've established a scholarship fund in his memory for travel scholarships for students. So that would have been an enormous loss if that freezer got emptied and nobody had deposited those materials. That's just one very visible anecdote, but we hear those things all the time. So even the communities that are sharing amongst themselves, once the lab is not in a lab anymore, there's nowhere to go for those materials. So thanks for asking. I feel like I got to talk a lot. Did I?
No, we just have a few minutes,
okay.
So that's great. That's because you're a speedy talker. Good for you.
Fantastic that this is doable in a private company, although not for profit. What happens to the repository you have if you fold?
Yeah, so hopefully we will never fold. So we have resources. We have, as a nonprofit, we're allowed to have money in the bank. It's not having money, it's what we do with the money. We don't have investors. We don't have, boondoggle trips to Mexico. We are actually shoring up investment to make sure that we can always maintain the repository. As far as the data, which I agree is often more valuable, we're working on a recovery, a plan for that. But right now, there's no, as long as there's a need for Addgene, hopefully Addgene will be here.
Let that be a closing remark. Good luck on that, and thank you so much, Joanne.